Okay, thank you everybody for coming. It's great to see so many people here in the audience. I seriously thought it was just going to be me and Frank and Janet and maybe a couple other people. So it's good to see that that people are interested in this. This session has come about because there's been some things in the Journal of American Oil Chemist Society and to some extent in AOCS journals in general, and we thought that, well, it'd be good to get some training for us about how to evaluate experimental designs in the manuscripts that come into our papers. I happen to know Frank through his participation, well, through Kraft and participation with our program at the University of Wisconsin, and he was great enough to say yes when we asked him to come and do this for us. So I'm really fortunate to be able to introduce Frank Rossi as a statistician at Kraft Foods, willing to share his knowledge and train us, so to speak, about how to evaluate experimental designs. Thank you, Frank.
Great, thank you. Good afternoon, everyone. First, just a little bit more about myself. I'm a statistician. I actually lead the statistics program at Kraft Foods. I've been doing that since maybe 2007. And I've been at Kraft Foods for 22 years. But actually, for most of the last 30 years, I've been a statistician in the food industry. I worked at General Foods Corporation. I worked at Campbell Soup Company. And there's actually only 3 years when I wasn't part of the food industry. And interestingly enough, and it's topical to this program today, I worked for the first statistical software company that was implementing designed experiments in software. So I'm of an age, unfortunately, that I actually learned to do all this stuff by hand. And it was in the late 80s that it was first implemented in software. And I think that's one of the reasons why we're here today, right? Software is relatively inexpensive to acquire, and it's easy to start playing around in it, and it's easy to generate things. And how do we know if it's good enough, right? So a lot of what we're going to talk about today is really, you can think of this presentation in two pieces. First is I'm going to talk about design of experiments, the statistical concepts, a lot of different aspects of this. And then I'm going to take about 7 papers that were either published or submitted to the journal. And then I'll kind of put my own criteria of what I think is, what I think is useful and important to have in a paper, and whether or not, in certain instances, this was delivered in the papers. The other thing is that, well, keep in mind 2 things. One is I'm a statistician and I'm not a chemist. Dad was a chemist, but I'm not. And so a lot of times I'm very good understanding the statistical aspects of the papers, but if we get into the actual chemistry part of it, that's where I would fall down pretty quickly. So you all have a much better knowledge about that if you're familiar with any of the papers. If any of the people are involved with any of the papers, it's not a criticism of your work. It's very much about what would we think should be included in a publication, if you will. And then the last piece is, as we go through this, feel free to ask any questions. So we've got a mic. The important thing is I'm being videotaped. And so because of that, this is for posterity, I guess. And what that also means is if there's going to be questions, we want to make sure that that gets in. So I'll try to also repeat the questions, but I think it's important to, if you have them, it's probably easier to address them right away as opposed to wait till the end, but I'll be happy to take questions at the end too. Yeah? So I'm going to start with just a very, very basic kind of concept, which is I think most people can get the general idea of baking a cake, right? So the formula itself has been determined, and we need to determine what's the ideal cooking time and temperature that's going to produce the best cake. And so we're going to vary temperature between 300 and 450 degrees Fahrenheit, time from 30 to 50 minutes. And the response here, how do I know what the best cake is? Well, our previous research has shown that a cake that has an internal temperature of 200 degrees is ideal. So it's not too dry, not too moist, it's just right. I made up the example, so I have no idea if 200 degrees is really good. If there's any baking people in the room, too. So our first step is we're going to bake some cakes where cooking time and temperature are systematically varied across the specified ranges. And then we're going to measure each for the internal temperature. And in this instance, the time and temperature combinations are going to form a square. A lot of designed experiment techniques are, if you think about it, there's a geometry to them. Squares and circles and cubes and hypercubes and spheres and things like that. And so my examples here are always going to be very simple in the sense that it's easy to view things in geometry with either 2 or 3 dimensions. But the concept applies whether I've got 6 or 7 or 8 dimensions. It's the same idea. And so when we talk about these being systematically varied, right, the corner points in this square represent the factorial combinations of the time and temperature endpoints, right? So the lower left-hand corner here, right, that's 300 degrees in temperature, and it's 30 minutes in time. Upper left corner is 450 degrees in temperature, 30 minutes in time. Upper right corner, 50 minutes, right, for 450 degrees, and then 50 minutes for 300 degrees. And then I've got something in the middle, right? And that's going to be kind of the midpoint of those. So it's 375 degrees for 40 minutes. Right? So it's all pretty straightforward? Yeah? Yes?
When you do not know your ranges, you can just arbitrarily define one?
Yeah, so, okay, as a statistician, right? Or maybe I should even say, put it a little more bluntly, the design doesn't care what ranges you pick. Technically, like when I learned to do these by hand, we just code them as -1 for the low value, +1 for the high value. Now, the success of your experiment depends greatly on having the right ranges. But that's not a statistical, that's not statistics. That's the chemistry or the baking science or whatever the topic that we're studying here. And so think about it as I've already done my work to know that this is the right ranges. And whatever you need to do to get there is very, very important. You know, I work with a lot of people doing product development experiments, right? We're making, say we're making cream cheese, right? They're going to make cream cheese in the pilot plant. They're going to make it over, they're going to make whatever type of design we ultimately determine. As a statistician, I have no idea if they've picked the right ranges for the factors. I have no idea if they picked the right factors. That's the word that a statistician uses for, in this case, time and temperature. So that's the food scientist or the chemical engineer or the person who can, ultimately is going to understand what the best range to pick is. As a statistician, I can jokingly say I don't care. I do. I want our experiments to be successful. But I certainly don't have the knowledge to know what the right ranges are. And that, the design is a design. It's a good if we do it right, it's a good design. But we can have a very unsuccessful experiment if we didn't pick the factors or the ranges for those factors very well. Does that make sense? Yeah. That's a good question, actually, because it really, often the success of something isn't about the design. It's about, did we pick the right factors? Did we pick them over the right ranges? Oop, heading in the wrong direction, right? So what have we learned so far? So I've just superimposed the actual measured temperatures in each of these, for each of these design points here. Right, so I'm trying to get to 200. So I kind of have an idea of the neighborhood, right, where I'm probably going to get something. Right, this one's 189, so not quite enough. This one's probably too much. These are kind of at the edges. So it looks like somewhere in here is where I'm going to get good enough. But at this point, I don't have enough data to more precisely identify this. And we'll talk about why in a couple of moments. So step 2 in this process, if I'm trying to get to that 200, is to bake some more cakes to better define the cooking time and temperature space, measure the internal temperature on these two. So now I've added these points here, right? This point, this point, this point, and this point. And these points form a circle, right? So think geometry again. I could have just picked them so that they formed a square. My preference is to push things out a little bit further, right? If I stretch over, right, a circle is going to let me predict over a wider space than the square. But there would be nothing wrong with, there's some very good reasons why sometimes I might choose to design this in a square. But I think you get the idea that I'm really framing the edges of this a lot better now. So these points are going to be sufficient to build a model to predict the internal temperature. So why did I run several more at the center? It's actually 3 more, not 2. Why would I have done that? It's certainly more work, and if I'm in the pilot plant at Kraft making cream cheese, I think you make a minimum 40-pound batch. Takes a lot of work, takes a lot of people, takes a lot of milk. So there's certainly work involved in it. Resources aren't free. Why would I potentially be doing more work? Well, 2 things here. One is it tells me something about the reliability here. I've made cakes at 40 degrees for 40 minutes at 375 degrees, and I've got very similar results for each of them. That tells me something about the reliability of my process here in making these, right? I've got to execute this, and I also have to measure it, right? There's a measurement system involved in this, and hopefully that's accurate too. So I want to see that these things are very similar. And remember, I've also added, right, I've done this in 2 stages. I did my initial points around the edges of the square, and then I added some points. And I want to make sure that, well, when I did this second round, that something hasn't substantially changed, and I'm adding apples and oranges together. I've got data that really shouldn't be put together because something has substantially changed. So that concept of doing some replication is very important here. Notice, right, the sphere, if you will, or the circle. The predictions for the model are only going to be valid within there. I haven't collected data at these extremes, and so I really have no idea what might happen there. Extrapolating is the term that a statistician would use, is not a good idea. We frown upon it very much. We consider it as like, well, gee, I'm using my model to predict in a space where I've never collected data. That's not a really good idea. Okay? So the math involved in this is something we'll talk about a little bit more detail as the presentation goes along. But ultimately, what that model does is let me develop an equation that lets me predict within this range. Notice I've changed my scale a little bit. I'm not going up to 500 anymore. I want to make sure that I'm able to predict within the space that I've developed my design over. But what I can see here is this line here, our model predicts any combination of time and temperature that falls along that line is going to reach an internal temperature of 200 degrees. So now I've got options here. And how would I determine what is the best combination of time and temperature to bake a cake? Well, I'd talk to the engineers and find out, well, what's the cost of, higher energy costs for running at a higher temperature? Obviously, a different time also affects throughput, and there's probably a way to determine what's the lowest cost combination of time and temperature along that line that's going to optimize my process if I'm going to use that as a comment. Yes?
Your first criteria was to go to cost, but what about whether different points in that line might yield different sensory attributes?
Yep.
Yes.
Yes. Yes, that's a very good point. So the point is, how do I decide amongst these? I use cost as an example, but there are many, many other criteria, and it's all very much based on the topic itself. So yeah, in this case, there may be, well, remember, you mentioned sensory properties, and so hopefully my ideal temperature of 200 relates to similar sensory things. But there could be many, many other things, and often we're experimenting, and we've got more than one critical response. I've used a very simple example. I've got one that matters, and then I can associate a cost to it. But really, often we have many, many responses, and we're trying to find the spot where they all converge to be close to the needed needed response range for each. And so often you're really forced into a very, very narrow set of combinations, or you have to trade things off one for another.
And that brings in to the first question that you wrote, is the versus square, the target point is why not 200 right away, and then determine the other parameters, because the main criteria for baking cake is the chemically, it's the what, the acquisition of the starch or whatnot.
Yeah. So I've used a very, very simple a very, very simple example here, really just to give you an idea of the concept. And there's a couple of key points on this that I want to make now. So first, it sounds funny the way to put it, but we don't really care about the individual experimental runs. I don't care that none of these things hit 200. And the reason being is we're going to use the set of them to understand the effects of time, and in this case, cook temperature, on internal temperature. Right? The design is created so that we can independently quantify the effects of time and temperature in the most efficient manner. And because of that, we need all the experimental runs. If I'm missing just one of these, this thing probably falls apart. It seriously compromises my ability to fit the model, generate those predictions. It sounds kind of funny because I've made like 10 or 12 of these. One missing could really make the whole thing fall apart, and the answer is yes. So that's a really key point here. And what happens, there are ways to deal with that. There are very complex ways to deal with it. But more often than not, if I've had a product developer call me up from the pilot plant and say, oh, I thought I could run that, but I can't, we're often in a scrambling mode to try and redesign things or come up with a point that's close to it that might not screw up the geometry. But think about that. If I don't have one of the corners here, I don't know what happens over there, and I'm not easily able to build a model that predicts what happens over there. So the whole idea of choosing your ranges, how you choose those ranges, maybe if you look at a design you've created and you think, I'm not sure if that one's going to work, you might do a little work up front to make sure it will work, because otherwise everything may fall apart, and then you've wasted a lot more time and energy. So, I've worked with people actually not just in the food industry, but for that 3 years that I worked for that software company, I got to see how people were doing these techniques in a number of industries. And one of the most important things is you need to do your homework up front. It's real easy for me to throw numbers up on here with time and temperature and all as a nice example. But there's a lot of work in getting to those things and getting it right, because we want to make sure we have all that data. Let's see, a couple other points here. We use the repeatability of those replicate runs as a ruler to assess the effects, the significance of the effects of cooking time and temperature. I'll demonstrate to you exactly what I mean by that in a couple more slides. Another thing that we didn't talk about here is the order of the runs. And we want to do this in a random way, ideally. And so we'll talk about what that means and what the implications might be if you don't do it that way. So those are two concepts that I really didn't talk about in detail at this point, but I will hopefully hammer them home pretty strongly at some point over the next hour probably. Because sometimes the design itself is great, right? Perfect statistically, but the details around how it was executed, right, can tell us something very much about, well, I might not feel as good about this when I just throw that thing up on a design matrix and it looks good from some standpoints. I don't know about other aspects that would tell me that this was done well or that it was done maybe not as well. So how are statistically designs different? Factors, and in my world, in the food industry, factors tend to be ingredient levels and or processing conditions. I think in your world, they're going to be other things. But that's the statistician's term. We systematically vary the factors so that the effects can be quantified in an efficient way. So efficient is a big part of this, because the idea is we're trying to not do more work than we need to. And that has implications too, and I'll talk about that. The statistical analysis of these studies focuses on the development of models that relate how the factors affect the responses. So that's why I say I don't care about the individual points. I don't care that none of them hit 200. I'm going to use the set of these to develop a model and use my model to get a better idea of what combinations of the factors are going to be ideal. Models provide a wide variety of information. So it could be just an ordering of important formulation and processing variables. So out of these 7 factors, which ones are the most important? Maybe some are more important than others. Usually that's going to be the case. It could be telling me an area of interest for further study, right? Maybe I didn't know up front 300, right? What do I have? 350 to 500 degrees and 30 to 50 minutes. So I might do some work up front to identify, oh, I really need to be operating at a higher temperature or a lower temperature. Optimal formulations. So in my case, in my world, it's usually products. So product formulations could be subject to cost and production constraints if applicable. And then ideally, you could even use these to define specifications, right? Not just what's the ideal thing, but based on if I knew that my temperature needs to be 200 degrees, but consumer tolerance of that, maybe 190 to 210, then I could determine, well, what combination of time and temperature is still going to give me my desired response within that tolerance, if you will. So there are several types of designed experiments, and they focus on different objectives. And so that's one of the crucial points here. Often when I talk to people about experiments, the first question I ask them is, well, what's the objective? And that has to be stated very, very clearly to be able to design an effective experiment. Screening designs are used to reduce the number of factors under investigation or identify factor ranges for further study. Right? Response surface designs, and sometimes people call these optimization designs, these are going to be used to develop a model of the design space that's defined by the factor ranges. So step 2 in my cake baking was a response surface design. Step 1 was actually a screening design. Mixture designs have factor levels that add to a fixed total, and their objective can be screening or optimization. So essentially, you have mixture screening designs, and you have mixture response surface or mixture optimization designs. One of the papers I'll show you has a mixture design in it. So when I talk about things that add up to a fixed total, right? Juice blends, right, are a blend of juices that add up to 100%. And I could be using 30% of juice A, 50% of juice B, 20% of juice C. They add up to 100%. What's the optimal mixture of those to get a certain characteristic? And again, you'll see an example closer to home in one of these papers, too. And often, in the Kraft world, right, we're dealing with products. I mentioned juice. Coffee is a blend of different bean types. We make mixed nuts. We make Planters nuts, right? So what's the optimal mixed nut blend? You've got your filberts and your cashews and your peanuts and the whatevers, right? Same idea. It's just things aren't independent anymore because they add up to 100%. And then I'm not sure if people are even familiar with this concept, but we call them robust designs. Taguchi was the person who really came up with the concept of using the design of experiments technique to look at determining factor settings that reduce variability to factors that are costly, difficult, or impossible to control. So in that instance, I'm designing an experiment to reduce variation. And I'll show you an example of what I mean by that. Yes?
I heard also that there were types called random design or random block design.
Yep.
How do they relate to these types of designs?
They're all related in the sense that there are factorial combinations involved in things. But an example of we'll say a complete block design, right? A randomized complete block design. Say I've got those 10 cakes, and we're all going to taste them. So I'm not going to measure them. Well, the measurement system is you people, and you people telling me how much you like them, right? I'm going to give you all 10 of these cakes, but I'm not going to give you each the same cake in the same order. How do I balance it so that cake number 1 is tasted the same number of times as the first cake, the second cake, the third cake, across my entire set of people? That would be an example of that. Or an incomplete block design that was randomized would say, all right, I've got 10 cakes, but each of you is not going to eat 10 different cakes. You're going to each eat 3 out of the 10. How do I make sure that the execution of how I serve the cakes to you is done in a way that doesn't introduce any kind of bias? So it's similar in a way in that there's concepts of structure and doing it in a very specific way, in a planned way. But it's a little bit different in the sense that the idea of factors being varied is a little bit different. They're often used for executional things. There are other examples in agriculture where that would also fit in too. So a screening design, so the concept of a design matrix is really the plan of the design. In this case, the columns, not in this case, in all cases in a design matrix, the columns are the factors and the rows are the runs. So in this example, I've got 8 factors in 16 runs. That's very efficient for studying these 8 factors. However, this is a screening design. I'm not going to be able to say what's the ideal combination of these 8 factors. Notice I've only got low and high levels for each of these. I made these examples up, and so I didn't even put any factor levels in them. But the idea, think about the structure being low and high factor level combinations. This is not randomized. I did that in very specifically to show you that it's not an accident that the first 8 rows in this design have the low level of that first factor, and the next 8 rows have the high level. There's going to be, in any column here, half of the rows are going to be at the low level, half of the rows are at the high level. It may not be apparent to look at that, but that is exactly the case. And it's very specifically constructed. This was not randomly constructed. It's specifically constructed so that I can look at each of these things individually and that they're independent or separate of each other. And I'll talk about that concept. Again, my cake baking design was also a screening design. Low and high level combinations at the corners, and in this instance I added what we call a center point. Right? That would be, in the parlance of that previous design matrix, that would be the 0, 0 point. Right? It's the mid-level of everything at the same time. And sometimes we do that, sometimes we don't. And there's very good reasons why we would or wouldn't in any instance. Response surface design. So think about one way to try and just conceptualize the difference between these two. That first design that I showed you had 8 columns and 16 rows. Right? A response surface design, in this case, I've got 5 factors. And I actually have 5 levels of each of those. You'll notice a +2 and a -2, a -2, +2 in each of these columns. So this is one where I've stretched the ranges in this case, a 5-dimensional sphere. So a response surface design tends to have smaller number of columns, a lot more rows than columns. Right? And in this instance, you'll see I've actually got 28 rows here, 28 runs, and then I've got 5 columns. Often, there are a number of common design types that are used. They have names. One is called, this happens to be a central composite design. There's another called a Box-Benken design. Sometimes people create custom designs. This whole idea of a de-optimal or a computer-generated design is one that is, years ago, well, before software, they didn't even exist. But there are ways to create designs that may not fit directly into one of the kind of the textbook types. Right? Central composite Box-Benken designs, I believe, have been around since the 40s and 50s. And so you would have to kind of force your problem onto the design. And a lot of times that's not a problem. But there are instances where I've got reasons why that doesn't quite work. So maybe the computer can help me generate a design. And again, 5 factors, in this case, 28 runs. This happens to be a central composite design. So central composite and Box-Benken designs tend to be cubes or spheres or some type of version of those. So my cake baking step 2 design, this is a circle, right? This is the minimum. There's 5 levels for each factor, by the way. So I've got 1, 2, 3, 4, 5 different levels of time, and I've also got 5 different levels, right? I've only got 1 at this extremely high temperature value, 1 at the extremely low temperature. This is the minimum required to fit a response surface model. So that's when I use the word efficient. It's because I don't need any more points than this to fit the model that I intend to fit. Now, that doesn't mean it might not be a bad idea to cover the space more densely. It wouldn't hurt to put some points in here. But if I'm making cream cheese in the pilot plant and I know that it costs me $1,000 for each run, I might think differently about how I cover things. If I'm doing experiments on the bench top and it's relatively easy and cheap, then I might cover that space more densely. But technically, all I need is to define the edge really well and to define the center pretty well to develop the model. That has implications about how you use the model, and we'll talk about that too. But ultimately, this is the minimum that I would need to do to fit a model at which I could predict throughout that entire circular space. I talked about mixture designs, and I used things like juice blends, but we could talk about something a little more fun here. If I'm going to make the ideal margarita, there is a Mexican restaurant here in the place, so we could do a little experiment after class if you want. Typically, a margarita, by the way, is 50% tequila, 25% triple sec, 25% lime juice. I could create a design around that space. Now, I wouldn't necessarily have one with 0 tequila, and I might not want to put a whole lot more than, what's my highest value here, 60%, right? After that, it might get a little bit much, right? Similarly, if I don't have enough tequila in it, maybe what's the point of having the drink? I don't know. Triple sec lime juice. So ultimately, I can, for 3 things that add up to 100%, I can represent them in what's called a ternary space, right? And so this is tequila along here, and this is 60% tequila, 20% triple sec, and 20% lime juice. So notice I've got a geometric structure here, right? And if I were doing juice blending, I could do the same thing. It's all about space and range. In this case, we've decided this is the right space to develop a model for the optimal margarita. But the concept would apply for anything else that adds up. Once you get into 4 or 5 or 6, it's hard to visualize these, but the same concept applies. So we've done, as I mentioned, right? You can do experiments with mixed nuts. I think there's 7 or 8 types of nuts in mixed nuts, depending on the specific product. And so the same concept applies. And I'm not going to have a mixed nut with only peanuts or only filberts, right? So I've got a constraint on those. I've got a minimum and maximum. But the same idea, think about geometry and space, and how do I cover that? This space now I've covered. It kind of looks like a square, right? Except it's kind of tilted because of that, the idea that they add up to 100%. You'll hear different design type names around here. They'll call them things like simplex and lattice. I won't go into the details around that, but those are the kind of names that you might see around that. When I talked about robustness, a very good way to think about this is, again, it's a different objective, right? The example I'll use here is, I think everyone's familiar that Kraft makes macaroni and cheese, and we actually have a product that's like a cheese in a pouch, and then dry macaroni, and you prepare them separately. You heat up the cheese, you you boil water, and you put your macaroni in that, and you put these things together to make a macaroni and cheese product. Well, I can't control how consumers are going to prepare this, right? They're not always going to follow the instructions perfectly. And I might want to make my product robust to some level of variation in consumer preparation. So if I overheat the sauce and underdrain the pasta, right, I'm going to have a very thin sauce. I've got pasta that has a lot of water on it. I put them together, I have a kind of soupy mess. On the other hand, the opposite of that is underheating the sauce and overdraining the pasta. So the pasta is very dry, it's cold. I put the sauce that has hardly been heated up enough together, and I've got a congealed mass of macaroni and cheese. Now, I can't control what consumers are going to do in their homes, but maybe if I change the ingredients in my formulation, I can make my product robust to that variation. Robust meaning it's not going to be as sensitive to the consumer preparation variation that I can't control. So it's a very, very different objective, but it's a very valid and important objective, right? And so often robustness is more important than anything else because there are things that I can't control or are very, very expensive to control, right? Sometimes think about things like weather-related type things, right? I either can't control the weather, or if I've got an old manufacturing facility, I guess I can throw a few $1 million into really good HVAC type controls to control temperature and humidity and something. But that external or that internal temperature in a manufacturing facility could be affecting what it is that I'm making. So this is you'll see this occasionally. I didn't find any papers when I was going through, but I could hardly say that I went through the last 25 years of AOCS journals anyway. So those are all the different design types. The analysis of this is now around fitting models. And I want to talk very much about how the models are, well, the models are used to relate design factors to responses. So their models are expressed in the form of an equation. An example is a very simple model for a 3-factor experiment, right? The x's are the factor settings, so these are factors, right? The betas are coefficients that are estimated based on the data from the experiment, right? A small beta indicates that the factor has a small effect on the response. A large beta indicates the factor has a large effect on the response. The sign of the beta indicates the direction of the effect, right? So if it's negative, it means as I increase this factor, the response decreases, right? So all of these designs that I've talked about are ultimately have some kind of underlying model that we're going to fit. And one of the, for a statistician, one of the very nice things about designed experiments is once you've designed it, you know what model you're fitting. There's really, you design it with a model in mind. And so rather if someone just comes to me with a lot of data, that's a little bit of a fishing expedition. I could try this, I could try that, well, let me see about this, right? That's a very time-consuming kind of thing. Whereas once you've designed an experiment, there's a model implicit with that, and that's the model that you're going to be fitting whenever the data is collected. So I could be designing something, and even if it takes 6 or 8 months for them to collect the data for whatever reason, I know what I'm going to do with that data when I eventually get it. Models can be very simple or much more complex. So first one was just what we call a main effects model, right? Individual effects for, in this case, 3 factors. More complex has main effects and what I'll call pairwise interactions. And I'll talk about that concept of an interaction because that's very important, but it's also something that we often build into our designs. So interactions. And then an even more complex is what we call a full quadratic model. So I've got my main effects, I've got my interactions, and then I've essentially got squared terms that help me to understand curvature, right? If I don't have squared terms, I'm assuming everything operates linearly, and in many systems that's not the case, right? The change in a response is different. It may be always increasing, but the increase is smaller at certain parts of a range and larger at other parts. So all of these things are linked, objectives, designs, and models, right? Designs are linked to project objectives, and then models are linked to the design type. So screening designs are going to be based on simpler models. Main effect models are ones containing some or all possible factor interaction pairs. Response surface designs are going to be based on more complex models, so full quadratic models, or when I say variance on those, there are some instances where, say you have a discontinuous factor, well, there's no squared term for that. But the same concept applies. I've got linear interaction and some type of curvature involved where appropriate. More data is needed to fit more complex models. So that's why when you saw more data means more runs. And that's why you saw when I had that screening design with 8 factors, I only had 16 runs. But when I had 5 factors for a response surface or optimization design, I had 28 runs. So I needed more data to fit a model with even fewer factors. The key to models that fit well and are easy to explain and understand is having a design that makes the main effects and the interactions independent of each other. And that concept is what drives the creation of the designs. It's all about making sure that I can keep things independent of each other so I know which ones are important and which ones aren't, and I can model the effects of them independently. So what does independent mean? Right? A very poor design, I'm just generically labeling these factors 1 and 2. Notice that with the low level of factor 1, I have the low level of factor 2. The medium level has the medium level of factor 2. Medium level of factor 1 has the medium level of factor 2. Or, I'm sorry, high level of factor 1 has the high level of factor 2. So I can't separate out the effects of these two because I'm increasing them together. They're not independent. The opposite of independent is the term we use. We say they're confounded with each other. They're mixed up with each other. And if, say, I see an increase in my response when I go from run 1 to 2 to 3, I really don't know if it's due to factor 1, factor 2, or maybe both of them. I simply can't tell. They're confounded with each other. And what we're trying to do is avoid confounding. So notice here, I've got what I call a better design where I've independently varied these. I've got, at the low level of factor 1, I've got the low and high level of factor 2. The high level of factor 1, I got the low and high level of factor 2. So I can separate the effects of these. They're no longer confounded. And the word we use is they're independent of each other. Now notice I had to make another run to do this. And so ultimately, independence means I sometimes might need to do a bit more work. But what you get out of it is a much clearer result. Yes?
Do you have an example where confounding would be helpful?
Would be?
Would be good to do?
I can't think of one, actually. Yeah. There are instances where we've confounded things. At this point in a 30-year career, I've designed a lot of experiments. And sometimes we do it because we're either trying to, like these things tend to move together in a manufacturing process anyway, and it makes no sense to separate them. Or I simply, I know that I'm going to have to meet other constraints, I put things together because otherwise it's not going to, it's not cream cheese anymore. It's an unidentifiable white dairy product, right? But so maybe, and this is where I fall apart because I'm not a chemist. I'm also not a product developer. I'm not a process engineer. But maybe if I vary homogenization pressure and mixing speed independently, I come up with stuff. Some of those things aren't cream cheese anymore, and that kind of falls out of the objective anyway. So there could be some instances there, but it's usually around those kind of things. So a full factorial design, which is all possible factor level combinations, guarantees that you'll be able to separate out the main effects and all pairwise interactions. In some instances, a fraction of the set of all possible factor level combinations will allow the separation of the main effects and the interactions. And we'll talk about, well, how would I know if I'm able to do that? There's a very specific way to tell. We can also use a fraction if fitting a simpler model is what's desired. And again, I'll repeat, if even just one design run is missing, the design may be compromised as the independence of the factors will almost, I should probably say surely be lost as opposed to just likely. So a design is characterized, the term we use is resolution. It relates to how complicated a model we can fit to the response data. So what we call a resolution 3 design, it says main effects are not confounded with other main effects, but they're confounded with pairwise interactions. So what that means is, say I have 3 factors, x1, x2, and x3. The main effect of x1 might be confounded with the interaction of x2 and x3. That if I have a lot of factors, I have a lot of pairwise interactions to consider, and that may be a strategy that's okay. I'm not going to generalize and say none of these are good or some are always good or bad. It's all situation specific. By the way, a statistician's response to every question starts with the two words, "it depends." And so that's, yeah, so we'll talk about these. And I've got resolution 4 designs where main effects are not confounded with other main effects, right, or pairwise interactions. The pairwise interactions are confounded with each other, right? And so that means I know my main effects are truly, those individual effects. The pairwise interactions I probably, I'm not so sure about. Resolution 5 designs are what we use for no confounding between main effects, between pairwise interactions, or between main effects and pairwise interactions. So the whole idea, and I'll talk about an interaction too, because I think the concept may not be clear yet either. But I'll talk about it so then you'll get an idea of why it might be important. So this is statistical terminology, but it's important in understanding what I can and can't get out of the design. So we use terms like to say, I have a resolution 3 design, and now we know what it means. A resolution 4 design is different, and I know what that means, and a resolution 5 design similarly. Yes?
Why did we start from 1 to 3?
It's a complicated answer, and it adds no value. So yeah, I know why, and if you want to ask afterwards, I can show you. But there's a reason, and it makes sense to me, because I know how these are generated by hand, and that's what it relates to. How you would actually create these designs. There's some multiplication involved, but it's probably not going to add value here because they come out of software. But if you want to stay afterwards, I can tell you. So often, this really comes down to a design strategy, right? It's a trade-off between available resources and information, right? If everything was cheap and easy and free, I would always run higher resolution designs. But designs that provide the most amount of information require the most amount of runs. Designs with smaller number may not provide the information we like. So an example here is I have 6 factors I want to look at. I want to investigate. It's a fancier term. I could do that in an 8-run resolution 3 design. I could do it in a 16-run resolution 4 design, or a 32-run resolution 5 design. Notice they basically double each time. That's the geometry. That's part of the whole aspect here, is fractions of things, right? There's rarely anything other than doubling to get to the next level, if you will. And sometimes doubling isn't even enough to go from resolution, say, 4 to 5. It depends on how many factors you have. And so that's what's beautiful about software is that once you understand the concepts, then that's the way the software talks about it, but then you kind of understand like what you're getting into and what your trade-offs are, what your choices are. And I think that's really an important consideration. There is no right or wrong here. It depends, and it depends on your situation. If you're shooting up space shuttles, 16 versus 8 might be pretty cost prohibitive, right? On the other hand, if you're doing something on the benchtop and it's cheap and easy to do and it's cheap and easy to measure, then why not do more to make sure that you get a better understanding of things? So we talked about, I mentioned replication and why it's important, right? It informs us about the reliability of the results. So my question here is hopefully obvious, right? Would you make the same conclusions about the results from both of these studies here? Look at the only difference here is the replicates. In the middle, 189 versus 191. Over here, 185 versus 205. The difference between those on the right is 20 units. And that's most of the difference between all of the points in here. That's half of the difference, right? The lowest is 165, the highest is 205. So the thing that's supposed to be pretty close to the same represents half of the range of my experimental data. That doesn't make me feel real comfortable about the reliability of this. In this instance, they're very close to each other, and that tells me something more. The factor effects here are much larger than the variability in the repeated runs. Factor effects here are not really larger than the variability in the repeated runs. If I go from here to here, that's the effect, one way of looking at the effect of increasing time at the high level of temperature. That's 20 units. That's the same range of those two repeated runs. So it's very important to build replication into a design if you can. And it tells you something about the reliability of your experiment. Now, if I got this, what would the problem be? Well, there's a couple things. First would be executionally, did we do this right? Right? If I'm a little sloppy, or in this case I could be more than a little sloppy, but that tells me something like maybe I should have done a little better to control things when I was executing this experiment. It could tell me something about the measurement system, right? No measurement system's perfect. That's a whole nother statistics topic I have passion about, if you will. But the idea here is maybe my measurement system isn't very accurate. Those two cakes were pretty similar, but my measurement system is not very reliable. I don't know what the answer is until we go delve into it, but I have another problem to sort out before I can think about doing anything with the data here in terms of my expected analysis. We talked about randomization, and that's the order in which we, well, make the experiment runs or execute them. That can be very important. So factors external to the design may influence the results. So I could have several batches of raw material that are needed to complete the design runs, right? And that may influence things. I don't know. But I want to make sure I don't mix that up with the things that I'm studying, the factors themselves. There could be just a time sequence effect from start to finish. So I've done a lot of the very first experiments I did actually were with coffee roasting, right? And when you're roasting something, you start up in the day and the roaster was cold, right? It does build up some heat over time. And I know that that's going to be the case. There's other ways to deal with it potentially. But if I randomize, at least I know that my little gradual temperature buildup in time isn't going to be mixed up with the things that I'm varying in my experiment. Raw material batches are another. We use milk a lot at Kraft, and milk varies. And sometimes if I'm experimenting a large enough experiment, I need different batches of milk, and I may need to worry about that. So randomizing helps keep the effects of any external factors separate from the factors in the experiment. So an 3-factor 8-run full factorial experiment. 4 runs can be made from each batch of a raw material, right? If I randomize the order here, the effect of raw material batches spread out across the main effects and interactions of these, right? So that's the point there. I know I'm going to need 2 batches. That's okay. But I just want to use randomization to make sure I don't mix up the batch effect with the things that I'm studying. Blocking is the term used when sometimes randomization isn't enough. I know that raw material effect is really big. I can't do anything about it. I wish I could run, the experiment with only 1 batch, but I can't make more than so many runs. So I need to account for it. So think about it now as that's a factor in my model. It's a nuisance factor. It's not one I really cared to study, but I need to account for it. So certain factors external to the design may have such strong effects they need to be considered. Randomization won't be enough. So including these factors as blocks means confounding the block levels with higher order factor interactions. I may need more runs to accommodate a blocking factor. My example here now, it looks similar to the previous one, but it's actually not. These are no longer randomized. This design was created with the batch effect as something that I'm going to put in the model. And because of that, in this case I was fortunate. I didn't need to do more work. But depending on the number of factors, the resolution of the design, I may actually have to do more work to accommodate that blocking factor. And so often people come to me and they say things like, oh, I know I'm going to run this in the pilot plant over 3 days, so I need to account for that. I want to block that, use a blocking factor. And I'm like, OK, well, tell me what's different about running it Monday to Tuesday to Wednesday. And I'll say, like, are we using all the same materials? Oh, yeah, same milk, same iron. OK. Same crew? Same actual physical assets to run this? Oh, yeah. It's just it's a different day, so we want to account for that. I'm like, well, do you want to do twice as much work? Because now you're going to have to experiment for 5 days instead of 3 or something. Oh, no. So you really need to understand, is that thing important before you go drive it into your experiment and do more work than you might need to. And maybe you need to do a little pre-work to understand if that thing should be accounted for or not. Because if you don't, it could really screw things up too. So all of this, right, the answer to everything is it depends. And doing your homework can make sure that you have a very, you get out of your experiment what you're hoping to. So in this case, here's where the difference is. Batch is confounded with the three-way interaction of factors 1, 2, and 3. So here's the cleverness to this, right? Multiply factors 1, 2, and 3 together. You get -1, you get -1, you get -1, you get -1. They're all batch 1. Multiply in this, the second, +1, +1, +1, +1. It wasn't accidental. It was constructed that way. Software helps with this, but that's what I had to do by hand. What used to take me the better part of a morning takes me about 10 minutes now, and it'd take less if I was a little better typing. So software is really amazing in this sense. But you still need to understand what you're getting out of it and how to get what you want out of it. Model fitting. So we're using regression analysis used to fit the design to the response data. Almost always that's what we're doing. There's a few instances where we might be doing something else, but every example that I saw in the journal articles that I looked at, the ones that we'll see today, are using pretty straightforward regression modeling. I don't know if you're familiar with regression modeling, but regression modeling gets really complex when things are not independent, right? Regression modeling is like really easy and very nice to work with when you've got this kind of a structure. So not only do I not know, not only do I know the model that I'm going to fit before I even have the data, I also know that the modeling is going to be pretty straightforward compared to just throwing a mess of data and trying to run a bunch of regression models and see what comes out. And that's part of the beauty of this too. So I've got another example here. The factors I'm varying are steam pressure, pump pressure, and line speed. My response is viscosity. I made all this up, by the way. The model for the design is the full quadratic model. So it's a response surface design. Hopefully I've designed it that way. I've got my main effects for steam pressure, pump pressure, and line speed, the pairwise interactions, right? And then the quadratic or squared terms for each of those three factors. So quick way to think about this too is the thing that increases these a lot, these models, the number of things that are factors, the number of things that you have to estimate in them is around the number of factors, and pairwise interactions are the things that pile up quite a bit. So if you have four factors, you have 6 pairwise interactions. If you have 5 factors, you have 10 pairwise interactions, right? It's always the number of factors times the number of factors minus 1 divided by 2. Always, always, always. 5 factors, 5 times 4 divided by 2 is 10 pairwise interactions. 6 factors, 6 times 5 divided by 2, 15 pairwise interactions. You can see how these models pile up in terms of the number of things. You need data for all those things. So that's why you need a lot more data when you're fitting these response surface models, especially when they have a lot of factors in them. Regression model results. So I'm gonna go into, the reason I'm going into this actually is because this is the kind of stuff that people put in papers. And so understanding a bit about what it means is important, but also how would you interpret them and what really needs to be in there I've got opinions about. So a lot of people have heard of R squared. It's the percentage of response variability explained by the model. So you want that number to be big, right? It can't be more than 1 or 100%, right? In this case, it's 96% or 0.96. So that's good. Is it good enough? It depends, right? Certain models should be more precise than others. If I'm working with, I don't know, I'll just use an example of food safety, right? I might want more precision than that because I need to be more precise. The total response variability is this sum of squares here. And then the response variability explained by the model is this model sum of squares. This number divided by this equals 0.960986. Always, always, always. Right? All of this stuff hangs together if you understand what you're looking at. This here is the significance test for the overall model. Right? In this case, smaller is more significant. Everyone's favorite cutoff is 0.05. There is nothing magic about that number. It's just a number that people often use, right? But smaller is better. It can never be 0. It'll truncate to 0 eventually in some software. And again, values less than 0.05 are often considered significant. Now, this is the actual parameters, those betas in my model, right? This is the estimate for beta 1. It's for steam pressure. Pump pressure, this is beta 2, 3. This is the interaction term, the interaction between beta 1 and, X1 and X2, steam pressure and pump pressure. This is the squared term for steam pressure here, steam pressure by steam pressure. My thing eventually starts to truncate the actual text, right? Notice something here. This is the way the software that I was using to develop this outputs it. But what's important to understand is they code the variables, the factors, back into that -1 to +1 range so that these are all in the same scale, right? Because notice line speed is in it's roughly a 1/10 of the scale of this. This is 22 to 40, this is 200 to 280, and 370 to 420. So they're different size of units, and so when we put them all back into a coded form, they're more easily comparable. Because if I look at these, right, I can see the associated p-values with each of these are going to be related to the size of them, right? And so the fact that this is minus 0.94, or minus 94, I'm sorry, is comparable to the two above it, even though they're in a different scale. Most software is going to do that. Some software will give you the equation back in the uncoded units so that you can use it to predict. So it's software's, they all have their own little nuance and stuff. And so it's important to understand what you're looking at because you need to try and make sure that what it is you're getting is what you're expecting, right? Notice each of these factor effects has a p-value associated with it. So the software that I was using here will eventually, it won't ever give you a 0. It just says it's very small, less than 0.0001. And then in this case, it's using a different color. Notice if it's below 0.05, it's putting them in red. If it's above, it's putting it in black. But 0.066 is a p-value that's pretty close to that cutoff of 0.05. And I would still think that maybe that's important enough in the model. Notice all the other factor effects are very, very high p-values, which indicate a high level of insignificance here. So I wouldn't fall on 0.05 as exactly the cutoff. Use your eyes. Realize, too, that in context with everything else, I think it's important. And in this case, I would still say that pump pressure squared is probably an important thing to keep in my model. Again, the significance tests, 0.05 again is people's favorite, but model fitting. To me, one of the most important parts of this process is not just fitting a model, but understanding what it means. So these interaction plots give us an indication of what's the effect of steam pressure. That's what's on the horizontal here. at the different line speeds and at the different pump pressures. This is the effect of pump pressure at different line speeds. So these two are the same interaction. They're just using, it depends on which factor I put on the horizontal and which one I break it out into different curves or lines for. So what this means is the effect of increasing steam pressure. Well, it's a decrease, but it's a smaller decrease at the higher pump pressure than at the lower pump pressure, right? The slope of this is certainly steeper at 370 than it is at 420. That was a significant interaction. And so an interaction means the effect of one factor is different at different levels of another. And that's often a really important finding. It means there's some type of synergistic effect or potentially even an antagonistic effect between those factors. Notice here there's no interaction between these. It's not that these lines are not one on top of another. It's that their slopes are more or less identical. I'm using the word slope a little bit cavalierly in the sense that these are curves, and normally we don't talk about slopes of curves, but the shape of those curves is almost identical regardless of whether I'm at 22 line speed or 40 line speed. So the interaction plot, I think, that's where a lot of the real results are. What happens when I increase steam pressure? I get a decrease in viscosity. But that decrease is different at different pump pressures. That's a really important finding. Notice on anything on the diagonal, this factor effect, this interaction is the same as this. It's just how I choose to graph them. This, the fact that these are so very separate from each other, is the factor effect of steam pressure, right? I get a higher viscosity at a lower steam pressure than I do at a higher steam pressure. So it's all the same information. It's viewed in a different way. But I think both can be informative. Now, you may choose one or other of the graphs to describe those factor effects, perhaps in a paper that you're going to be trying to publish.
Is there software if you cannot control precisely the pressure, for example, but you can measure it precisely?
Yeah.
So you have one of the runs instead of 200 at 205, the
Yep.
other at 195?
So ideally, your design is going to be based on things that you can control. So my first piece of advice would be to say, all right, are there things that you can control that effectively change that factor? So for instance, often we're interested in varying pH. pH is a hard thing to vary, but if I change something with the acid levels, is that effectively going to give me a change in pH? Now my model would be based on acid level, right? And so we understand that as I change that factor, there's a change in pH, and that may be what's driving the factor effect too. So in general, you want to design around factors that you can control and control pretty well. And the real problem would be if they're so difficult to control that my low level, my medium, and my high level are starting to get very close to each other because they need to be as separate as possible to keep that independence. So my general response would be to caution you against trying to do it that way because I've seen it fail more often than I've seen it succeed. And if you were to do that, I would say spread them very, very far apart. So if they vary a little bit, they're not likely to overlap or come close to the other levels in the design. Model assessment is another important aspect here. And what that means is the residuals in a model, that's the term we use for the difference between the observed points, the ones that I have measurements for, and what the model predicts. And the assumption that my model is a good model suggests first that they fall in a normal distribution in some way. So I've got a histogram of them here. They need to be centered around 0. They should look, this looks fairly bell-shaped here. Nothing's perfect. They fall around a normal probability plot, so nothing's deviating from that line. There should be no patterns in these. This is versus the fitted value. I wouldn't want to see larger residuals at the ends of this than I would be, say, towards one end versus another. So I don't see larger residuals at either end of this. Versus the order, right? This is the order in which these things were run in. So these are all parts of assessing my model. So it's not just looking at R squared. That tells you something, but it doesn't tell me about. All my residuals may be small, but maybe they don't follow a normal distribution. Maybe they're not equally, some roughly equally, some negative, some positive. Maybe they're different at different levels of the response variable, and maybe there's some relationship with the run order. All of those would be watch-outs to suggest maybe there's something more in this model that I'm not accounting for well. Model refinement is the term that we use when people may decide to remove the insignificant effects from a model, right? Simpler models are better and easier to explain. The idea of parsimonious models is the word statisticians often use to talk about models that we've removed all of the insignificant terms from. So I'll start by saying this is not as critical for designed experiments. And the reason being is designs are created so the factors are independent of each other. The conclusions of factor significance will not change very much. Model predictions will be very similar for full models and refined models where I've taken out the insignificant effects. So is it necessary? I'll say that it's preferred, but I will acknowledge that depending on the software you're using, it may not make it easy to do this, right? So sometimes software makes it difficult to work with, especially multiple responses. Reason being is I've got a different model for response A, viscosity, than I do for response B, water activity. I've got different terms that were significant, and software sometimes says, I want you to use the same model for all your responses because that's the way I've implemented this. And so that's why I say it's preferred, but I will acknowledge that in some instances, given the software that people have available, it may make it difficult to do some of the other things that are important, which is I want to be able to look at all my responses and see where they converge to that ideal. Now, why did I say that it's maybe not as critical for a designed experiment? So here's my full model here. Remember, I've got these three terms that are insignificant. I've removed those three, and that's my refined model. So look at the estimate for steam pressure, 265.14. In the refined model, it's 265 minus 265.14. It's identical. What about line speed, minus 94.72, 94.72? Pump pressure squared, 78.12, 76.19. Very similar, not identical, right? So my conclusions aren't going to vary much. Interestingly enough, though, notice this p-value for pump pressure squared. It was 0.06 before. In the refined model, it's actually smaller. The significance of these is actually higher levels of significance. Smaller p-values mean higher levels of significance. So one advantage of refinement is that, again, this thing that didn't maybe look so significant, now I might have a somewhat different conclusion about. And that's related to kind of removing some of the insignificance in models. And it affects the test, the significance test for effects. So that's where you'll see, and that's another reason that even if your software forces you to use the same model for all your responses, you may think a little differently about that 0.06. By refining the model, I've shown that its p-value is less than 0.05. It tells you something about the, I don't want to say arbitrariness of 0.05 as a cutoff, but I'll say the subjectiveness of the cutoff of 0.05. Is this point clear about refinement? Yes? So if you have decent software, the energy and time involved is minimal. Yeah. And so to me, investment in a good piece of software is well worth it, because, it might cost you $1,000 or $1,500, but if you're doing this a lot, then the time that you're saving to get, and be able to work with these designs. And it also includes it has to do a lot with all those other things like the refinement tools, the assessment things, like how do I look at my residuals and things like that. A decent piece of software will make all of that very easy for you to do. And if that's the case, then it's probably well worth the investment. So, like I said, the creation piece, I know how to do all this by hand. I've done it all by hand. And when I got out of graduate school, we didn't have software to do it. So now, like I said, if I can design something in what used to take me about 2 to 2.5 hours and do it in about 5 to 10 minutes, that's certainly a point made there about the utility of software and worth the investment. Model verification. I think this is a really, really key step. And when you see, when we look at and critique a few papers, I think it's the place you're going to see that often has not been done at all or not very well at all. Why is it a critical step? Remember, I didn't cover the space densely at all. I covered the edges and I covered the middle. So that leaves a lot of white space in there. And I don't know if my model might look good statistically, but boy, I should make some runs at my ideal condition to verify that I am really getting the response. My model is a good predictor. I'm going to get the response that I would want. So notice here, in this case, I was trying to get to a viscosity in this example of 4,300. I have a lot of options here. At line speed 22, it's all around that curve. At line speed 31, it's around this curve. At line speed 40, it's around this curve. So I got a lot of different options here. And very few of them are going to be design points. It's very possible none of them will be design points that I've actually run. So I need to understand, well, is my model going to predict well? Now, I don't need to verify every possible thing that I might be interested in. But to verify, you need to run factor level combinations, measure the responses, and then compare the model predictions to verify the model's accuracy. Model verification is helped when some of the original runs from the experiment are repeated, right? Because say my model predictions don't match my actual observed results. Well, remember, I ran these verification runs days, weeks, months later. And again, that whole thing of, well, if I run something now and I run it tomorrow, how do I know I haven't controlled things well, right? So if I repeat runs from the original design space. So this example here shows that I've got 6 runs from my original design. I got my original viscosity. I've got my formula prediction. And then hopefully my verification viscosities are going to come close to these. That tells me I didn't have a problem in executing because I did this again 2 months later and I'm not putting apples and oranges together. These runs here are things that I never ran before. These are ones that I care about because I'm trying to get to around 4,300. Right? Notice I chose 6 here that have a pretty wide range in viscosity. I'm going from around 4,061 all the way up to almost 5,000. So I've picked things that are going to be different to help make sure that my verification, my model predicts well over a wide range. It doesn't just predict well at my target of 4,300, but it predicts all over the range because maybe my target will change later. These are all at this point, my desired condition. They're all around 4,300. Notice they're very different. I've got different steam pressures here. I've got different pump pressures. They're all things that I pulled off of that graph. And hopefully, the ideal here would be my verification viscosities come very close to the predicted formulas, the observed when I've got them. Otherwise, just the predicted ones from my model here. Yes?
So which criteria did you use to pull out those values? Again, the cost concept, the ones on the bottom?
The ones on the bottom? Yes, I could use cost. I could use whatever's of interest to me here. So this is a completely made up example, so I didn't really think about it that way. But the idea would be, yeah, maybe running at different line speeds is very important. Sometimes I know I'm going to run at a higher or lower depending on the throughput that I need, right? I need to build up sales volumes or something like that. So if I say this is barbecue sauce, right? I make more barbecue sauce in the warmer weather months. We sell more. So I need to run at higher line speeds. I still want to get to my target viscosity. So that was the concept that I had in here. But it is completely made up, so. I have to make them up if I'm talking in public, by the way. So again, maybe differences are not the model, but the second execution. That's why repeating some of the original design points. Now, how many is good here? It depends, right? Cost, all those other things. But not doing it all at all is a real problematic in my mind. So now we're going to spend, actually, let me stop for a second and say, because we're moving to the next piece, which is we're going to look at a bunch of papers. We're going to apply some criteria around this. But any questions on any of the stuff that I've gone through so far? Because we got about 35 minutes. Yes?
My question is about replications.
Yes.
If we go to the example, you showed us the square plot.
Yep.
There was one which had 20-point variation and one that had 2-point variation.
Mm-hmm.
So certainly, if there is a 20-point variation, we want to go to a third replication. But if the variation is just 2-point, is it OK to do duplicates? And if yes, then how do we represent the data? Averages and the standard deviation or standard error, what should we represent?
So that's a good question, actually. And I think part of this is, remember, you probably won't know what that replication variation is until you've completed your experiment. So you kind of have to decide up front how many replicates you're going to have. And that, I think, is both a resource question, and I'll show you an example where people have done some interesting things. So to me, I think, unfortunately, the question is, I'm not going to know until I run the experiment. It really tells me more about, now that I've run this, do I think it's going to be useful to me? Or do I think I need to rethink the execution or the measurement of this, get that straight, and unfortunately repeat it because the replication variation is so wide? Did that answer the question?
Duplicates is enough to do if we decide as a team.
Yep.
We decide that duplicates are OK.
Mm-hmm.
How do I represent that? How do I present that data? We cannot do standard deviation on a duplicate, but how do I present it?
I fully believe that, if I've run a design and it has 28 runs, wherever the replicates may be, that I want to see all of the 28 runs represent in a paper. Yeah, and I was actually I've been critical of it when I've seen other papers. So, I mean, I understand that's what people do often if they're presenting information, but in this instance, just seeing how big of difference those numbers are is very important, and just throwing a standard deviation off it, I think, can sometimes, I don't want to say misrepresent it. I think it isn't as clear what level of variation I have in the replicates. So I would prefer they all be just put as individual runs. And maybe, I think it's OK to order the presentation table in a way so that you can see all the replicates together, even if they weren't run in that order. Yeah? I'll show you an example where I think they've done a good job of it in the way in which they present the experiment. Other questions? Yeah? So if the lack of fit is significant, what that means is my model doesn't fit particularly well. I may be able to talk about some of the factor effects, but the ability to, because they could be significant still, but what it's saying is my model is probably not going to predict very accurately. And so if I see that, it would suggest I don't really want to use it to make predictions. I can still talk about what we've learned, because in some instances we say, oh, increasing the steam pressure does this, but I can't be very precise in explaining. So increasing steam pressure decreases viscosity. That may be a fairly good statement to make, but I don't want to be precise about increasing from 200 to 250 is going to give me a decrease in viscosity of 482 centipoids. Okay? Sure. Other questions? Okay, so we've got some papers here, and here's what we're going to do to judge them. Things to consider first about the design. So does the design match the objective or the stated conclusion? So again, you can't optimize with a screening design. You need at least 3 levels for each continuous factor for optimization. You need to be able to fit at minimum the full quadratic model for optimization. How was the design executed? Randomization or a restricted randomization would be best. So I didn't mention explicitly, but a restricted randomization would be, is done, and it's often a reasonable compromise. So when I mentioned coffee roasting, right? Every time you change the roaster temperature, well, actually, you may as well go have a cup of coffee because it takes about 30 minutes to get to the new temperature set point. So you don't want to make a lot of changes in temperature. So you might do like a block randomization, so I minimize the number of times I change temperature, but I still randomize the other factors within that. That's a reasonable compromise, and it might mean that instead of taking 4 days to execute my experiment, I could do it in a day. That's a very reasonable and a very practical thing to do. So, but people need to be explicit about that. That's what they did. If not, is there a compelling reason why you wouldn't randomize? And I have a hard time coming up with a compelling reason. I haven't seen one yet, but I want to at least open up the possibility. This should be mentioned or discussed somewhere in the text of the paper. And is there replication? Because again, this helps demonstrate the validity of the results. Things to consider about the analysis. How much of the analysis is shared in the paper? So have they actually put the design and the response data into the paper? Do I see ANOVA or regression output? And the subsequent p-values? Have we visualized the factor effects, and how have we done that in a way that people can truly understand what we've learned from the experiment? Is it clear that the authors understand the output in the analysis? Is there a focus on just 1 or 2 things in the output, like an R-squared value and a p-value? Is there any assessment of the models and how well they fit the data? So is there any of those residual analyses? Plot of observed by predicted is often used. Have the models been refined? So again, the significant, insignificant terms removed from the model. And then is there any verification of the model's ability to predict? So we're going to use those criteria on this score sheet. And there's 7 different papers that we're going to talk through here. And I've just taken each paper's got maybe 2 or 3, at most 4 slides, where I've actually taken parts of the output to show you what was demonstrated within this. So, and then we're going to use the criteria on the left. So there's 1, 2, 3, 4, 7 criteria, and we're going to rate it, this is my rating, I guess, as good, fair, poor, or in some instances, it just may not even be present. So I have no way of judging. So this first one, I'm not going to try and pronounce author names because they're complex. But basically, it was a 2006 paper, Optimization of Enzymatic Alcoholysis of Soybean Oil. This is where we're like way out of my area of expertise. I might not even want to mention the titles anymore. So in this case, the design, it's a face-centered cube central composite design. 3 center point replicates, so I would say this is an appropriate design for optimization. Again, they used optimization in the title. Notice that they've given us within this the observed response data, the predictions, and the residuals are included in the paper. There is no discussion of the design execution, so nothing about randomization that was present in the text. It doesn't mean they didn't, but I think it's important to list in there, right? The statistical analysis results include model coefficients and p-values. So in this case, there really is just I should go back for just a second. So these are the 3 factors: temperature, enzyme or oil weight, enzyme to oil weight ratio, and substrate molar ratio. They've got an observed methyl ester percent weight and a predicted. So it's really just one response here, and optimizing based on that. They give us the p-values. Associated with all these, you'll see that all of our factors are highly significant, very small p-values. R squared is 0.90. R squared adjusted is really used for comparing models with different levels of refinement, so there's nothing wrong with including it here, but it doesn't really add any utility because it's used to compare different models. So the authors have refined the model by removing statistically insignificant terms. So notice there's only 2 interactions here and 1 quadratic term, and there's 3 factors. And then the model results are visualized in contour plots. Only the barest assessment of fit in the model is given with this plot of the observed by the predicted. So again, the residuals are the difference between the points and the line. So if the model fit perfectly, all of the observed and predicted would fall within that line. Again, the R squared, in this case, is listed 0.96. That's probably on the original model before refinement. No validation of the predicted optimum. There's no validation of the predicted optimum. So they list a predicted optimum. They don't validate it by actually creating and doing more experimental runs. And it's actually out of the design space. So the optimum level for enzyme to oil rate ratio is listed as 0.09. The range of this in the design is 0.1 to 0.2. So they've identified an optimum, they've gone outside or extrapolated, and they never actually verified it. So my rating for this paper, if any of you are in the audience, this is my rating only. I like the design. It's very appropriate for what they're trying to do. The replication was sufficient. No evidence. I really can't judge one way or another, but I would hope to see randomization, and I think it's important to mention it in the paper. I thought the output presentation was quite good. And the output discussion, that's kind of hard to summarize that in the paper, but I thought they did a good job of discussing the output in the text of the paper. Model assessment, I put in poor because they only did that one observed by the predicted plot. I would like to see more there in terms of, does it follow a normal distribution? Are the residuals centered around 0? All of these things, is there equal numbers above and below 0? Is there any time sequence effect? Is the size of the residual dependent on the response value? So I didn't see any of those. So they did something, which is better than nothing, but that gives it a P instead of an N. And then model validation, again, they did nothing technically to validate it. They predict an optimum. Again, they predict it outside of the range, but they don't actually do anything other than use that prediction, and I think it does need to be verified. Example number 2 here, incorporation of stearic acid into, I might have truncated some part of the title. Okay, palm oil and palm kernel oil. Okay. So again, design here is a 3-factor central composite design, this time in a sphere. Previous one was a cube. Both are fine. 9 center point replicates. I did the oh my god thing there. I was like, well, I mean, considering the design has 15 design points before you do any replication, and they've gone up, and put 9 in there. That's a lot of extra work to do. And I'm not saying don't do it, but it's probably more than was necessary. What was really interesting in the text here, notice they're very true to it. They've got the response data here, and it's actually just blank for 2 of the design points. So digging into the text, it says 2 design runs were excluded from model fitting. They explain them as just outliers. They didn't really give any discussion of why, so I don't know what the data points were. They just decided, I guess, they didn't like them enough. Maybe they truly were, but to eliminate this and go through and continue with the modeling, especially because these are points on the edge of that sphere, really does compromise the modeling. Again, they're critical for fitting the model. But they did go ahead with this. No mention of randomization. Statistical analysis does include model coefficients and p-values. Probably hard to read them when they're at the, e to the minus 0.008, but authors have refined the model by removing statistically insignificant terms. The model results are visualized in contour plots. So I like that aspect. Model assessment, they did a normal probability plot of the residuals. They also have that observed by predicted plot. So they did a little bit more than the previous paper in terms of assessment. They did it's funny. They actually did a very nice validation. They did 5 additional runs. The expected versus the observed. They actually did a chi-square test to say whether or not the set of points, the observed from the expected, are significantly different. Chi-squared at 1.8 for 5 or 4 degrees of freedom is not significant. So this was actually, so all of these, frankly, are going to be a mixed bag. I should probably say that up front. That in this one, my biggest criticism is this sort of like, oh, we didn't like these 2 points, but we're just going to go ahead anyway, without a whole lot of explanation in the text. It's critical to the model fitting. But then so many other steps in this process, they did quite well, actually. And this is maybe one of the better validations I've seen in a lot of papers that I've looked at. Any questions, comments? Oh, yes?
Shouldn't the software say an error when you have missing data?
No. Ultimately, if you can't fit the model, if you don't have enough data, you will certainly notice from the output. But software isn't there to be the police, so to speak. Because again, remember, regression analysis isn't always used for just a design. It's used for any set of data. And so the software truly can't do a very good job at policing you anyway. So my overall rating here, appropriate design, poor because of those missing points and no real. So the design was originally good, but kind of going past without having those design points makes me go back to poor. Good replication, no mention of randomization. I like the presentation output. I like the output discussion in the text. Model assessment was a little better than the last, so I give it a fair, and model verification was very good. Example 3, optimization of yeah, right. I'll let you guys do that. This is your area now. This is one where they've used 2-squared full factorial design with center points at each of 3 levels of a third factor. This is not an appropriate design for a true optimization since curvature cannot be fit for 2 of the 3 factors. So I thought it a very odd way to construct a design when, frankly, you could do a similar amount of work with just a standard 3-factor central composite design or box-Benken design. Replication and randomization were included in the design. Notice they've given us the design points, the order in which they were run. So even within the table, it's obvious that they randomized the order. And then they've given us all the response data, which I like when you've got a number of responses and you can get an idea of how, especially when you get to replicates. Notice they do have replication within here, and you get a feel for how true those replicates are to each other. Statistical analysis results include model coefficients and p-values. There's no evidence that any model assessment at all was performed. So no observed by predicted, no normal probability plot of residuals, nothing at all within the text of the paper. There is a detailed discussion of the modeling results, and they're visualized in a number of informative plots. The authors use and description of the desirability functions to assess multiple responses demonstrates understanding of their modeling. So interestingly enough, even though the design wasn't particularly the right design, multiple response optimization is not a trivial thing. They've got 4 responses. And the way in which it's often done are what they call desirability functions, in which you say, what's the desirable response for each of these? What's the best result for each of these responses? And then it creates a function and helps you to find where that sweet spot is, where they all get to their ideal. And the way in which they discuss it in there, they truly understood what they were doing with that, which is not trivial. So I was pleased to see that. There's no validation, though, of the optimized conditions. So again, they come up with optimum, but then they don't kind of go to that next step. So my ratings here, appropriate design is poor, good for replication, good for randomization, output presentation and discussion is good, no model assessment at all, no validation. So notice I changed my rating to this. I said good because the design was good. I should have done that in the previous slide. Somewhere in here I had a change of heart, right? And it was poor because, again, there's missing points and just sort of like a lack of acknowledging it other than to say we took them out. This is an interesting one. I pulled this out because this is actually, it says experimental design applied for cost and efficiency of antioxidants and biodiesel. They actually run a mixture design here. The design points here are, it's called a simplex centroid design. It's very appropriate for mixture optimization. They've gone to the full range of this. So given the three different antioxidants, they've used either all of one, 50-50 blends, or 1/3, 1/3, 1/3, a tri-mixture they call it. And then the control is actually not part, it's kind of like biodiesel with no antioxidants. So that's kind of like we're trying to do better than this or see if the antioxidants help, I guess. They replicate the center point, that's included in the design. The center point in this case is that tri-blend, by the way. There's no mention of randomization within this. And notice they've used cost here, right? Antioxidant cost. So, by adding them, I guess there's an additional cost to it. Is there a benefit versus that cost is really the purpose of the study. Only very basic analysis results are presented in the paper. They don't really talk about the factor effects. They just talk about the overall model, the fact that it's significant, and there's no mention of model assessment or refinement. I will say that modeling in the mixture designs is not, it's kind of complicated, though it's a little more complicated than just a straightforward response surface design, but it's not that much more complicated to just not do it any more than this. And again, no mention of either assessment or refinement. Visualization of the response surface in a three-dimensional plot is more sexy than informative. So yeah, it looks kind of cool, doesn't it, right? This is your ternary space here, and they projected the response. Kind of hard to get that, well, maybe it's sort of easy enough to say this blend, when I've only got one antioxidant, I get this low response, I get the top one, but it's kind of hard to project that. A contour plot, which can be done in that ternary space very easily by software. If your software can do this, it can do the contour plot. So I kind of was like, yeah, I think they were going for sex appeal there, if you call sex appeal a contour plot in the world of statistics. The authors do a nice job of describing in the text the factor effects and their implications of both the response of most interest and the cost. So they understood what they were looking at. I think they could have done a better job of presenting it. The authors use multi-response optimizer functionality in the software to determine a cost-effective blend. So here's cost. Here's the response, right? They're trying to maximize the response here. But there's no validation of the blend, though they refer to that the prediction's close to an observed response in the design point nearby. So that's kind of like a poor man's optimization. It's saying, well, this one's kind of close to that point where I ran, so it's probably, that validates kind of. I think you could do the exact thing. But I will acknowledge that they did at least mention that. So for this one, good design, fair level of replication. They did kind of the minimum. No mention of randomization. Output presentation was pretty basic, so I call that fair. Good output discussion, no mention of model assessment, and again, poor for model validation since they really didn't create anything additional. They just said, well, it's close to that. Yeah, I'm not even going with this one. It is a box-spanking experimental design. Box-spanking design limits the area where the model can be used to predict. Now, that may be okay if they don't use the model to predict out there. But a box-spanking design, if you think in three factors, and they've done in this case, they've varied four things. But in three factors, think about that cube, the design space being defined as a cube. A box-spanking design would place a sphere within the cube. So you can't get to the extremes in the corners of the cube. Your model isn't going to be able to predict there. Now, that's okay if you don't use the model to predict there. But let's see if we get that. There is no mention of randomization. In this one here, there's no statistical analysis results presented in the paper, only the regression equation. So you really, the equation itself looks to be in coded units for the factors, so the coefficients are in similar scale, though the factors themselves are not. There's no evidence of statistical assessment of the significance of the factor effects. I have no p-values, I have no r squared. I have no way of knowing if this model fits well, and it may, or it doesn't fit at all. I just simply don't know. All they've given us is the equation. Some indication that it fits well is in their, what I say, only the barest assessment of the fit of the model. They plot the observed by the predicted. The residuals are the difference between the points in the line. Your r squared is 0.9935. So that would indicate the model does fit well, but I still don't know which of these factor effects are important. Factor effects, interactions, I don't know what the nature of those are. I just have an equation. Discussion of the factor effects is based only on the visual evaluation of the factor effects from viewing three-dimensional plots of the response surface. So again, maybe a little more sex appeal than helpful. Again, main effect and interaction plots would make it easier for readers to understand the nature of the factors and their interactions. So you remember I had my kind of matrix of those interaction plots. That's sufficient to really understand, you know, which of these factors has what kind of effect on the response. And then the optimum, though, in this case is validated with 3 replication runs. So again, almost every paper's got some good and some maybe less desirable things. So my assessment, appropriate design is fair. I didn't get a feel within this. Oh, I should say, the reason I say fair is they've used their model to project outside of the design space, the sphere within the hypercube. They have a 4-dimensional space here. So I'm not real happy about that, but they didn't really talk like they were predicting out there. So it's kind of a little mixed. So I gave it a fair for the design. It is a good design, but I see that maybe they don't understand that they can't predict where it can and can't predict in. But I don't have a good feeling either way, I suppose. Replication was good. No mention of randomization. Output presentation is poor. I mean, there's very little. Output discussion in the paper was very poor. There was almost none. Model assessment, again, they did something, that one plot. But then they did a nice job of validation. So this one here, I'm not, esterification of sanitation waste for biodiesel. All right. 2-factor central composite design. They call it a 3x3 factorial, which is also correct, and it is appropriate for optimization. There's no mention of randomization here. So good design, and then they've got different responses here. The analysis of variance results for all responses are provided in the paper. It's evident the authors have refined the models by removing non-significant terms, and they provide the final prediction equation. So I was pleased with that. You can see what the significant factor effects. This was for one of the responses. They have similar tables for the other. There is no assessment of the fits of the model. They don't even tell us the r squared. I mean, everyone tells you r squared. So we don't really know how accurately the equations are going to predict. Identified optimum values were all design points, so it appears the developed models were not used to determine them. In other words, they just kind of eyeballed the data and they said, well, this one's supposed to be good for this, and this looks pretty good for that. And again, remember, the design points are there to define the space. So there probably is a true optimum for these that's somewhere within that, you know, in this case, it's just a square. But all they've done is say, well, here, this one was close to the corner one, and this one's close to this other point. And so it's not really using the design and the analysis to really identify true optimums. Also, there appears to be an error in this, since 40 is outside the design space, but there is a data point at temp and 60. Remember, they did give us the data, corresponds to this value. It also neglects to mention that responses at the replicate points are 91.9 and 92.2. So yeah, I would have preferred that they actually gave us the individual replicates, I guess was my point with that. Visualization of the response. Everyone loves these plots, and Go ahead. I mean, include them, but give me the other ones because I can't. I've been doing this stuff for 30 years. I have a hard time reading that. So again, a contour plot's going to be more useful. Discussion of the factor effects is based only on the visual evaluation of the factors by viewing these. Main effect and interaction plots would make it easier. So a recurring theme from me is that visualization could be done with some simpler plots. Include these if you like. I don't have a problem with it, but if that's all you're using, it's a little hard to follow, I think. So again, good for appropriate design and replication. No mention of randomization. Output presentation and discussion is only fair. No real model assessment. And then model validation is kind of poor because they really just kind of point to design points in that. But I guess that's better than doing nothing. Last example here. This is relatively new. Notice some of these go back a ways. So optimization of mixing parameters. I'll leave the rest of it. Three-factor central composite design in a sphere is appropriate for optimization. Though not explicitly mentioned in the paper, the design execution appears to be randomized. And observed and predicted response values are included in the table. So I like that. ANOVA results for all responses are provided in the paper. It is evident the authors have refined the models by removing non-significant terms, and they provide final prediction equations. So I thought that was pretty good. The discussion of the model assessment through R², a lack of fit test, and observed versus predicted plots. So a little bit more than we've seen for assessment from some of the others. Contour plots and multi-response optimization plots. So they're overlaying the different responses within this design space to kind of come up with an overall, like here's how we got to our optimum level. Here's where these things converge. I'm not going to go into all the details of the plot. They're a little complicated to read, but it's very obvious that they understood what they were looking at. They utilized their models very well to kind of find the space where their multiple responses were optimized. And then the predicted optimum was validated with an additional experimental run. So they actually went out, they said this is the good thing, they went out and made it, and then discussed in the text that the results were close to what the model was predicting. So for this one gets good for many things: design, replication, randomization, output presentation, discussion. I would have liked to see a little more in model assessment. I would just call it fair, and also fair for they did verify. They did the one run. Might have been good to try and replicate some of the actual design runs again, but they got the results that they wanted, so I suppose that's pretty good. So this was really my kind of view of a number of papers. I tried to get a wide variety of things here. Again, if you look at these, no one's doing horribly in every way. It's more a matter of there's certain themes, I think. Not a lot of mention of randomization. Maybe people are doing it, maybe not. But I think it's important to note in here. Assessment is maybe not very well done in most instances. And also validation is there's a couple of good instances, but often it's either not evident they did any or it's done rather poorly. And I think that's where I'd like to see a lot more of this in any of these papers going forward. We still have time for a few questions, I think. So I'm happy to answer anything. I talk fast sometimes, too. I live in Chicago, so we talk fast. But so any questions at all on anything I've discussed here? I think we're going to...
Yeah, I have a question. I should perhaps mention that I'm working for the publisher who has published these papers.
Okay.
Is, from your assessment, anything that you would consider flawed? Or is it just, it could have been done better, but it's okay as it is published?
Okay, so the one with the missing design points, that's to me a big deal to have just kind of blown off in there. On the other hand, the rest of that paper shows a lot of sophistication. So I can't say that any one of these is so horrible, but what I would hope is that we would go back and during this review process to say, all right, tell me what happened there. And maybe they did something that was reasonable. I just have no way of knowing in that instance. So I've rarely seen anything that was so egregious that it was just, you know, can't believe this ever got to print. But there's a lot that I think that we should maybe be very consistent about expectations for people going forward, and things that I do think that most papers in this, with these kind of designs, should be demonstrating. Any other questions? I'm like standing between you and a really nice day. So, yes?
For model assessment, I know you mentioned there were 4 different things. I only got 2 of them. There was like the normal probability of residuals,
Yep.
observers who predicted the R-squared thing, and there were 2 others.
Yeah, let's go back to that. We'll get there. Oop, there we go. So, yeah, a normal probability plot is just a way to, possibly even just the histogram is good enough to show they're centered around 0, have roughly equal numbers, positive and negative, and that they follow sort of like a bell-shaped curve. Similarly, I want no patterns in the residual plots. So in this lower left corner, it's versus the fitted values, right? And then versus the order in which they were run. So that shows that there's not any bias throughout the execution of the experiment, that maybe I get higher residuals at the later runs during that phase.
All right, so the one in the upper left there, then, that's the observers who predicted the R-squared?
No, this is a normal probability plot. So it's a way of assessing whether data falls along a normal distribution. So the scale on that, if you look at it, it's a very unique scale. It's a way in which you can plot data, and if they fall pretty close to that line, then it's a good heuristic way of saying the data follows a normal distribution. If I had points, especially, you'd see them at the ends often that are very far away from the line. That would suggest that the data does not necessarily fall along a normal distribution.
So the R-squared associated with that line is not the R-squared associated with the fit of the model?
That's right. They will not likely give an R-squared with that at all. And it's true that observed by predicted is something that I saw in a lot of papers and is not typically what, in the world of statistics, we would use as a way to assess the model. It's informative in some ways, but it doesn't give me all the information I need, whereas these are really giving me a lot more of that information. So I thought it actually a little bit curious. It never occurred to me to plot observed by predicted and say that that's model assessment. It seems very interesting that it comes up a lot in this field, and I'm not entirely sure why, but it gives me some information, but it doesn't give me the requisite information that I would need to be convinced that the model's truly assessed well.
So that's the difference between a scientist and a statistician, because I would plot a residual plot too and want to see how far they, or the comparison plot.
I was
see how
fairly
far-
certain statisticians are scientists, but maybe not. Yeah, yes.
We will find it acceptable in the paper they to say we did these spots and we discovered no anomalies, or you think this should be included in the paper?
My preference would be it's to be included. Now, that being said, right, I'm being completely, I don't want to say ignorant, but not cognizant of things like, right, more graphs and things probably increases production costs and other things like that. So I would leave it up to the editorial staff to decide if it's mentioned in the text, is that good enough, or do I need to see pictures? In general, my preference would be to see pictures.
How we determine the number of experiments that we had to make in order to come up with a valid conclusion?
Yep.
Is there any formula, rule of thumb, or there is a magic number?
There isn't a magic number, but I would typically say this. So when I said validation, right, I said two things. I said, I want to see some repeated, that's not a good thing to stand in front of that, some points that are original repeats of the original design, and some that are predictions at, responses that I care about. Now, I've got 6 of each here, which is possibly more than what I think most people would do. So then you have to, I would like to see a discussion of why did I choose to do maybe only one of each. You know, if the cost of this is very dear, I think that's a reasonable thing, and you've kind of done the minimum you need to do, given that. If you're trying to predict over a pretty wide range of responses, and you have a number of responses, so I don't just care about 4,300, but I care about other response levels too, then I'd want to see validation at those too. So I guess the answer is it depends. You knew that. But I would say at minimum doing one of each would be kind of the absolute minimum that I would expect. And if that's all that's done, then I would hope there'd be an acknowledgement as to why we've done that minimum level.
So let me expand on that question, because we sometimes get the comment that, well, you did 8 experiments to generate 19 different beta factors.
Yes.
And so you can fit anything with that, right? So maybe a different way to ask the question is, well, how many experiments would you have had to run to adequately fit 19 beta factors?
Well, you need more than 19. And the real question isn't how many of those, but the structure of them. That's the whole point of the design.
Right.
And so I would expect to see a very standard, classic design used. Or if it's one of these computer-generated designs, then there should be something to, and it gets into something that's a little beyond what we were talking today, but with computer-generated designs, you can assess the level of independence that's within those. And because sometimes people generate something that's the minimum amount of data sufficient to fit the model, but they're losing a lot of the independence and all those characteristics that I said were important for a good design. And there's ways to show that, but I would need to see that in a paper if someone was using a design like that, because you wouldn't get it by just looking at it. But if someone uses a phrase like, I've got a central composite design, then that tells me that I should have sufficient data to fit that model, that it is the minimum sufficient amount of data, but it should be sufficient for it.
Other questions? If not, let's thank Frank for that wonderful workshop.
Thanks, Stu.
Thank you for attending.